Welcome, everybody. This paper I've been looking at for a little while now. I've heard about layer imaging. This is the first time I read a little bit deeper into the paper and got acquainted with what Huber and his colleagues have been doing. Layer imaging concept has been around for a while. The idea is the brain is composed of distinct layers in the neocortex, about 6. And also in certain areas, especially the visual cortex, it can be organized into columns. So at higher field strengths, higher spatial resolution, looking at a smaller region with fewer slices, could we possibly resolve the activity and the structure better within the layers themselves? And if we take a look at something like Free Surfer, just to motivate why looking at a finer resolution is such a good idea in general, when we have large voxels, let's say 3 by 3 by 3 roughly, this voxel here is especially large, but imagine that the underlying brain is the most faithful representation of the finer grained details, and my voxel here might be active, but it could be a smearing across different regions. Okay, so think of these different gyri as a placeholder for different layers, different columns, for example. So if I see the voxels active, is it because of region A shaded in green, region B shaded in yellow, or is it some mixture of both? So it can be very hard to, or maybe impossible to disentangle this. And in a somewhat different connection, people have called this a partial voluming effect. When they talk about partial voluming, usually they mean one voxel can overlap 2 or more tissue types. Think of a voxel that might be on the border of, let's say, the corpus callosum and the ventricle, something like that. It may be mixing the signal across 2 separate tissue types, but it's also a problem with fMRI results as well. And that's why if we do something a little bit more sophisticated, like we reconstruct a 3-dimensional volume into a 2-dimensional surface, you get a better idea of where the activation follows the natural folding of the gyri and the sulci, which is how we know in reality the brain is organized. So here you can see on the right, is our volumetric results, peak T statistic for just a sample contrast. This is incongruent minus congruent is about 4.44. And on the left, if you resample it to the surface, first of all, you get better localization of where exactly the activation is, and the T statistic also improves, probably reflecting the fact that things like smoothing, averaging across nearby areas, isn't smoothing across functionally heterogeneous areas or possibly different tissue types. This approach, reasonably, is probably more powerful. It's more physiologically plausible. And the idea behind layer fMRI is similar. We're trying to get to an even finer spatial resolution so we can resolve these very small but neurobiologically important differences within the brain. And here are some data from, I'm sorry. Did somebody have a question? I thought I heard something. Here is a study by Goenza and Logothetis. Logothetis did a lot of work early on establishing that the bold response really is closely connected to underlying neural activity, which gave us more faith that the bold response is a reliable epiphenomenon of actual neural firing. Here we're looking at different slices focusing on the visual cortex. This is within the monkey brain, anesthetized monkeys. And you can see percent signal change at very high resolution. This is very high epi resolution, functional resolution. Gives you a better sense of where the activation hugs the gyri. Now, it's not perfect. I believe this is a gradient echo sequence. It can be a little bit more sensitive to field inhomogeneities. You also can see that within something like the calcarine fissure and extra tissue areas, cerebrospinal areas where there might be draining veins, for example, it can pick up these bold signal differences that aren't really the same thing as neural activity. It's farther downstream. So this is a so-called nearby draining effect, which can lead to spurious activation where there really is no underlying neural activity. This has been a problem we've known about since the beginning when we're talking about neurovascular coupling. How do the neurons signal that more blood is needed? And when this oxygenated blood is filtered out, there can be this massive influx of oxygenated blood, and some of it just tends to go through the waste pipes, filtered back into the CSF to be recycled. And this in turn can lead to some spurious, not really spurious bold effects. It's still a bold effect, but kind of spurious brain activity effects where we wouldn't expect to see it. Here again, if we focus on things like differences between gradient echo and spin echo, just a brief summary of the difference between them from MR physics. You may recall that spin echo, it uses a rephasing radio frequency pulse. So you tilt the hydrogen protons, let's say 90 degrees, and there's going to be some random interactions. Some of them are going to start spinning faster, others might be spinning a little bit slower. Then you apply a refocusing pulse, let's say 180 degrees, like flipping a pancake. Now the faster ones are behind, the slower ones are ahead, and they tend to converge into a signal that averages together, and you're able to get a stronger signal from that. A gradient echo, on the other hand, uses a single radio frequency pulse, but uses phasing and rephasing gradients to then try to establish a stronger signal that you can then acquire. So gradient echo, and the physicists in the room, throw me some rope if I'm maybe confusing some of these topics, but gradient echo will allow you for much quicker TRs and TEs. So in general, this can be used for accelerated neuroimaging, but again, can be much more sensitive to field inhomogeneities. So what you're seeing on, I believe let me see here. Let's make sure I have this right. Forget which one is spin echo, which one's gradient echo, but one of these is more sensitive to veining drain effects. I believe the one on the left is gradient echo, and the one on the right is spin echo. So gradient echo, it can be more sensitive to actual effects, but it can also be more sensitive to artifacts as well, which is one of the drawbacks. Now, specificity is very important when we talk about layer fMRI because, like I said, typically human neocortex has 6 distinct layers, and we categorize these usually by the histology. Now, this is a very famous figure by Senta-Gotay back in 1978, a drawing showing the distinct layers. You see the Roman numerals 1 through 6, and also different types of layers. There's pyramidal cells, there are so-called chandelier cells, there are Martinotti cells, there's microgliiform cells, many different types. So we usually categorize these different layers by their histological properties, what kind of cells there are, and how they're organized. This is a great idea, but there are many issues with layer fMRI. So before anybody plans to conduct their own layer fMRI experiment, be warned, caveat emptor, so to speak. One is that layer fMRI, looking at these really high resolution voxels, is going to be much, much noisier than conventional fMRI data. If you remember from an intro fMRI course, you may recall that the noise that we see in a typical fMRI scan can be as large, maybe even larger than the fMRI signal. And if you get to a very high resolution, it tends to be dominated by thermal noise and random noise from the scanner. And you also have to worry still about things like motion. So how are we going to account for that? We'll talk about that later on in the talk. Second, this can require, it really necessitates almost local smoothing. Remember that smoothing with a typical fMRI pipeline, you take an average, a weighted average of a voxel and its neighbors to try to cancel out noise and boost signal. It's a very reliable way to increase signal to noise ratio. But even then, even at 2 by 2 by 2, 3 by 3 by 3, you can possibly start to average across functionally heterogeneous areas or maybe different tissue types, and that's no good. You're essentially averaging noise together or contrasting signals, which what's the difference at that point? Local smoothing, on the other hand, is much more restricted. There's a command in AFNI called 3D blur in mask, for example, which can take a gray matter mask or an ROI you're interested in and only apply smoothing to that specific region or that particular tissue type. until you get to a desired smoothness. So it's much more accurate. There's less leakage of the signal and the smoothing across different tissue types or different layers, as the case may be. So in the case of something like Layer, fMRI, and the LayNII package in particular, they really emphasize local smoothing, let's say within columns, if you're focused on that, or along the layers. And there are different parameters you can set to make that specific. The last issue is because if you want higher spatial resolution, remember there's always this trade-off, really no matter how strong your magnet is. We don't have the technology yet to solve this puzzle. But the trade-off is as spatial resolution goes up, temporal resolution is going to go down. If you want finer temporal resolution, usually that means you have fewer slices, so your spatial resolution is going to suffer. Now, when we talk about layer fMRI, usually the studies are going to have restricted coverage. If you have sub-millimeter or maybe even micrometer resolution, probably just for your own sanity and the sanity of the subject, if you're scanning a live human subject, they're not going to be in there forever, and you'll need to pick and choose exactly how many slices you want in order to acquire the data set in a reasonable amount of time. They summarize this in a somewhat droll figure. If you look at the labels of these different axes, how well does a certain kind of pre-processing pipeline work for the majority of people? So unacceptable, that doesn't work really well. Something like smoothing, isometric smoothing, for most people works relatively well, even if everybody does it the same way. Everybody smooths at 6 millimeters for a typical fMRI data set. We accept that. It works pretty well for cortical, subcortical areas, so pretty good across the board. You start getting into much more customized and bespoke operations, which is really required for something as specific as layer fMRI. You're going to be seeing that the same kinds of pipelines is not going to work very well for all people. So this is, again, speaking back to the drawbacks of layer fMRI, you need to be prepared to be spending a lot of time customizing the pipeline for individual subjects and for your study. For example, gray matter segmentation with layer accuracy usually requires some degree of manual intervention. It's kind of like FreeSurfer, but actually a lot more demanding. FreeSurfer, you try to account for any discrepancies in the gray matter reconstruction, the peel surface, for example. This goes beyond that because we're talking about individual layers of very fine resolution. You want to make sure that the gray matter segmentation is close to perfect. In case people don't like graphs like these, they much more food oriented like I am. Think about it like if you're trying to satisfy a lot of people, you can give them all McDonald's cheeseburgers and it'll be okay in general for maybe some or most of the people. You want to satisfy everybody though. You give them these customized burgers that look really, really delicious, depending on the people's appetites and tastes. If they're vegetarian, you can give them Beyond Burger. So let's get into the details about how layer fMRI works. Let's take a really well-behaving gyrus and sulcus. So here's the gray matter, the cerebrospinal fluid's outside of it, the white matter is interior. We're going to call the voxels that are on the surface of the gray matter and white matter, on that interface, we'll call those the inner gray matter surface. And the ones that touch the interface of the cerebrospinal fluid, we'll call those the outer gray matter surface voxels. Now, within this gray matter folding, which we know has a certain curvature, a certain shape, these involutions that we know are the characteristic, really the defining feature of Cortex that makes it so compact and efficient. These can be distinguished into layers, divided into layers. So you could have 3 layers, you might have 2 layers. This is one of the options of the user. And you can make it more than 6 layers if you want. There's really no restriction on it. I mean, if you have not great high resolution, like let's say you have like 0.3 millimeters as opposed to 0.1, you might not want to use that many layers. We'll see some of the differences later on. Conversely, you may be also interested in columns, and not all columns are going to be equal depending on where they are along the curvature. If it's a straight shot from the inner gray matter surface to the outer gray matter surface, these are very straightforward. But if you're along the curvature, it may seem like a pyramid or a triangle with the tip cut off. Okay? One of the principles about this, which is very interesting, I didn't know about it until I read this paper, but apparently about 100 years ago, 1929, there was a Dutch neuroanatomist named Siegfried Bok, B-O-K. And what he found was there appears to be this equi-volume principle that governs the brain structure. So within the gray matter, what tends to happen, it's not as though this thickness looks the exact same across all the foldings, but it does appear that as you go closer to the crown of a gyrus, the more outer parts of it tend to get a little bit thinner, and the parts closer to the white matter get a little bit thicker, and vice versa as you go down into the depths of a sulcus. So the volume across the entire winding path of this gray matter ribbon tends to be pretty much preserved, which I find fascinating. And apparently this has been validated over the years. His observations have been confirmed. It's not going to be like exact, but it's a pretty good approximation, which, they try to respect in this particular software package when they reconstruct the gray matter. When we try to create columns or layers, as the case may be, first of all, let's say that your highest resolution are these voxels right here in this discrete image. And we'll resample this. We'll talk about these being the actual, say, like for sure gray matter. These are the ones in green. The ones that touch the interface of the CSF, those are outer gray matter surface voxels. The ones that touch the white matter are the inner gray matter surface voxels. And the algorithm they use tries to make the distance between the outer and the inner gray matter voxels as short as possible to reconstruct layers or columns. So I'm going to direct your attention to this part of the figure, this cortical depth values. So let's say, for the sake of argument, the resolution of our voxels is 1 millimeter. So one voxel equals 1 millimeter. The distance here from the outer gray matter surface to the inner gray matter surface is 1 to 3 millimeters. Very easy if it's a straight shot from one to the other. When we get to, let's say, this voxel to this voxel, what's the quickest path? Well, obviously, it should be a diagonal. And what's again saying that these are 1 millimeter isotropic voxels through the Pythagorean theorem, which I haven't used in a while, but glad I learned it in high school. That means that this hypotenuse, this diagonal bisecting this square, is going to be square root of 2. So the overall distance is 1 plus square root of 2. And if you want to make it compact, 2 times square root of 2, because these are two diagonals, plus 1. So they create these things called streamlines. Similar idea if you've used diffusion imaging like MR tricks, these streamlines to try to find the most efficient path from the inner to the outer gray matter surface. And then it uses another algorithm to determine what these belong to, how to organize these in neurobiologically plausible columns and layers. So I hope that makes sense. If anybody has any questions, just let me know. But it took me a little bit, but once I figured out this was the Pythagorean theorem, it made a lot more sense. So keeping in mind how these streamlines track the cortical depth along this gray matter, they have this equi-volume metric. They also try to make however many layers you reconstruct, try to make the distance between the layers also equal. And if you put this onto, let's say, the big brain, this is a very high resolution brain acquired by Amunds et al., I think published back in 2013, if I'm not mistaken. So here, 100 micrometer isotropic resolution, so quite high. And if you apply this layer fMRI approach to it, the LayNII. So first pass, it does this segmentation and also estimation of the layers and the columns. You can see along these bulges how the ones that are bulging out get a little bit more weighting as closer to the outer GM, for example, in this case. And maybe in the sulci, there's more weighting closer to the inner white matter surface. Equidistance and equivolume don't always track perfectly. You can see these arrows, there's some difference between them. Equidistance tends to be relatively stable across the entire ribbon. Equivolume can have much greater changes. And when you start to create as many layers as you want, again, remember that both of these principles are going to try to be adhered to by their software. I mentioned earlier that the resolution can also have an effect on how smooth these layers are, how well they reconstruct them. And I haven't collected my own high field, high resolution data yet, but they do have data online you can play with. And for example, let's say that if you try to do layerification and resample it to a 0.4 millimeter grid, there's nothing stopping you from resampling to a higher resolution than the native epi. Just so you know, it actually tends to work okay in that case. So here you can see it's actually relatively smooth. And let's assume that we've tried to reconstruct 3 layers here. So it does a pretty good job at reconstructing all of them. If you try to do it at the native acquisition size, it's a little bit more jagged. Here you may lose a connecting voxel between one of the layers. Doesn't seem to work as well, might be a little bit more neurobiologically less plausible. And here, if you have anisotropic, it might suffer from a certain dimension, right? So the dimension that is the lowest resolution is not going to be reconstructed as well. But a lot of this can be addressed possibly by resampling it to a finer spatial grid. Let's say that you're trying to estimate columns. Let's say you're interested in the visual cortex or part of the motor cortex, whatever you want. Their algorithm is going to try to reconstruct it into a desired number of columns. And again, this is a parameter you also have some control over. So you might want to base it on previous literature or what you know about the particular anatomical area. So if it's a closer match, it's probably going to do a better job at reconstructing exactly how many columns there are. This was taken, I believe, from an anesthetized cat. And here's the original convoluted cortex. And you can see in this structured columnar area of the cortex, there's some pretty clear lines dividing the different columns. If we unwrap it as though it's just going from left to right, more like kind of a flat sheet, you can see the distance between these different columns. And this can also be represented in, say, like a two-dimensional sheet of the central sulcus in this case, which can extract them and give you a good idea exactly what the distance is between these different columns, how they shade in one to the other. And from there, ideally, if you're then looking at fMRI results on top of this, it can give you a much finer, much more accurate representation of things like finger movements in the central sulcus bordering on the motor cortex. A few other options that are available in the LayNII package are selectivity. So smoothing, I mentioned before, they recommend doing some kind of localized smoothing that may go along the layer or along the column, for example. And selectivity is going to restrict that amount of smoothing based on how many nearby voxels are within a certain variance range. So I believe, I'm testing my memory here, but I think something like 0.04, 0.08, that's the percentage of nearby voxels that are below a certain variance threshold, which seem to be very similar to the central voxel, which is the center of the smoothing kernel at a given time. In this figure, they show that if we have a source voxel here, we smooth, say, along one of the layers. The selectivity, if we make it a little bit lower, becomes a little bit more a little bit weaker. It's possibly going to be smoothing across areas that are maybe not as functionally homogeneous. But if we bump up the selectivity a little bit to 8%, they become a little bit smoother, and you can see them track a longer area of the layer. I don't have data for what the difference is between, say, false positives, false negatives for this, but it seems to be around a selectivity 0.08 from their figure, tends to work pretty well. They also have a so-called no kissing option. And what I was showing in some of my first slides was a voxel that bridges multiple gyri, for example. And here you can still have the same issue, even at very high resolution. Some of these gyri are packed very tightly together, and you risk having a certain resolution element that might bridge both of them, leading to signal leakage across them. So the no kissing option will try to take into account nearby anatomical information to try to separate these as far as possible, assuming that they are two separate regions. Here in the bottom left, they have this matrix of different parameters, where different smoothing kernels from 0.5 to 4 millimeters. Also, whether the no kissing flag is off or on. This is tracking cerebral blood volume. We'll get into the difference between that and bold in a little bit. But let's say, for example, let's assume there actually is two separate peaks. There are two separate peaks in two separate anatomical locations, two separate gyri. So the profile should probably look something like this, where you have one sharp peak here, then nothing as you go into the sulcus, and another sharp peak over here. If you have a larger smoothing kernel and you don't use the no kissing option, trying to prevent any kind of leakage across layers, the distinction between the peaks tends to go away. If you have it on, the peaks get a little bit more differentiated. And also, if we look at something like the selectivity parameters combined with no kissing, it tends to do an even better job at resolving the peaks within these different layers. The authors also mention how quality assurance checks should be done when applied specifically to layer fMRI, and they compare it to the traditional quality assurance checks that you would use for an fMRI data set. So up top, these first four panels show you what we usually look for in a typical fMRI data set. Over here, if we look at the underlying epi image, it should be looking like a brain. Temporal signal to noise ratio should, usually I've heard be around 100 if possible. Here it's going up to 40. And statistical activation scores, let's say it's a visual checkerboard activation task, those statistics should be pretty high and also confined, relatively localized to the area that you think they should be in. So visual activity should probably be restricted to the visual cortex and not bleeding over into CSF, other areas of the brain, especially after correcting for multiple comparisons. Compared to that, the QA checks for layer fMRI look more at the profile of the noise, because we know that the noise is much more predominant at very high resolutions, and especially in layer fMRI. So their advice is, if you look at the distribution of noise, which should be roughly normally distributed in most cases, you want the skew to be close to 0, so pretty normally distributed. The kurtosis, the lower we can have that peak of the bell curve, is also good. The correlation with, autocorrelation from different voxels should usually only be elevated within gray matter. We do know that, for example, with the bold signal, there is some temporal autocorrelation. You can make a pretty good guess what the signal is going to look like at one time point if you know what the activity is from its neighbors. That's the definition of autocorrelation. And here you can see, if we have a couple areas called out, highlighted in cyan and also red, you don't see any issues really in the metrics for an fMRI data set, but with layer fMRI, something like The kurtosis is very noticeable in areas like this. Also, the skew, you see a clear difference over here, but you don't see any noticeable differences in the QH checks with a typical fMRI data set. They also mentioned something called VASO and compare it to a typical bold response. VASO, think of it as inversely related to cerebral blood flow. The idea behind VASO is trying to eliminate some of these draining vein effects, some of these vascular effects that you may not be specifically interested in, and they use a blood nulling pulse sequence for that. So if we know the T1 weight of the blood, and if it's distinguished from, say, the tissue itself, we can have a pretty good idea where there's more tissue activation as opposed to maybe activation that's being spuriously located in the veins. If we look at a flashing checkerboard task, for example, the bold response over here in the bottom left is going to be very clear. It's a very good bold map, but you can also see maybe some of it is starting to get into one of the draining veins. Something like VASO, it's a little bit counterintuitive. VASO is typically looked at in terms of percent change going down, negative percent change. Again, think of it as 1 minus cerebral blood volume. Just the way that it's set up, it's trying to remove some of these bold effects as though they were contamination almost of underlying tissue activity. You can see there's a pretty strong anti-correlation between the bold signal and also the VASO signal variability. So you don't see as wide a spread of these activation maps or deactivation maps as the case may be, but it does seem to be a lot more spatially specific and probably controls better for false positives. So this is one of the choices you have when analyzing layer-specific fMRI data.
Can I jump in on there for a second?
Yeah.
So VASO is it's a really cool technique because what it does is it essentially makes it so that all the signal from the blood goes away. And usually you collect these images with a spin echo so that none of the T2 star effects show up.
Yes.
So you lose the sensitivity to oxygenation, but pretty much the contrast that you see is that whenever the blood volume goes down, the signal goes down. So what happens is that this kind of takes away that weighting that happens on the veins with the bold effect. So the bold effect is largely, it's blood volume, but it's also about the oxygenation level. So when you do VASO, you're kind of doing away with the oxygenation. So you don't really see the veins changing anymore because the venous volume doesn't change so much during activation. It's mostly in the arteries and the arterioles. So it kind of takes away that draining vein effect. So it's almost like necessary if you want to do layer-specific fMRI to do something that's not bold. Like you were saying this earlier, right?
Yeah.
And it's because the VASO is like really the key to, or there's other techniques that you can do, but essentially sort of the key to doing this. So anyway, I just kind of wanted to add a little bit.
Thank you. Thanks for that clarification. I know that this is more your wheelhouse. And also, if I'm not mistaken, you can combine a gradient echo sequence with VASO, correct? So gradient echo can be more sensitive to certain changes in activity, but VASO can also eliminate these draining.
Yeah, you can combine them, but I think in general it's probably I mean you can do gradient echo, but the idea is usually when you do VASOs because you're trying to get rid of the bold effect.
Yeah.
Right. So if you're going to do that, then you want to do gradient echo, but you want to shrink that echo time so that you can reduce that T2 star dependence.
Well, thank you. In the same vein, pardon the pun, they also mentioned how you can devein, like try to really account for these draining vein effects using different models. Now, I'm not sure, and it doesn't seem like they're entirely sure either in the paper which one is superior, but there are a few different models that are out there. trying to account for what the physiology might be of these draining vein effects. It does seem in general, as you get to the more superficial layers of the cortex, there appears to be a stronger draining effect. Just the way that the angiography of the brain, the way that the blood vessels and veins are outlined, you tend to get larger veins, larger vein effects, the closer you get to the surface of the brain. So one model is a so-called linear offset. We just assume that as you go from, say, the inner surface gray matter voxels to the outer surface, you're trying to account for the fact that there might be these vein effects and then try to weight them accordingly. Scaling assumes that this is more of a multiplicative effect. It's not as simple as just a linear scale going from one layer to the other, but it actually seems to have these nonlinear properties. And lastly, macrovascular leakage assumes that there might be, again, some kind of interactions between different layers. As you go from one layer to the other, maybe some of the signals bleeding into one and the other. This also has some interactions possibly with the smoothing artifacts from different layers, even though LayNII will try to keep those as separate as possible. So here we see a few different maps suggesting that, if you use linear, this can give you a higher signal to noise ratio. It might be physiologically a little bit more unreasonable given what we know about the vasculature of the brain. If you use, let's say, leakage deconvolution, the signal to noise ratio can go down, but this does tend to be a physiologically more plausible model of how the veinage and the veins and the drainage in the brain interact. Again, I'll leave it up to you which one you want to use. I haven't seen a direct comparison which one is the best, maybe for controlling for both type 1, type 2 errors, but you have different options. To summarize, LayNII, they really emphasize offering customized analysis pipelines. Again, they don't want a one-size-fits-all. They talk about how you're probably going to be doing a lot of manual intervention, especially during segmentation, especially during looking at different amounts of layers, columns for different kinds of analyses, and also what kind of models you want to use and acquisition parameters, pulse sequences, to minimize draining vein effects. At higher fields, the bonus of all this, why we would go through all this trouble, is you should be able to get layer-specific resolution at higher fields, and possibly even at lower fields like 3 Tesla. That remains to be seen. But at 7 Tesla and above, if you adequately control for these sources of noise, and the segmentation is accurate between gray matter, white matter, CSF, this should give you a much more localized picture of which layers are activated during the task, for example. Now, there's ongoing work for deveining and denoising models. Again, combinations of maybe spin echo, gradient echo with VASO, for example, are certain ways to account for these veining effects. And denoising models also, because we're so dominated in layer fMRI by thermal noise, by scan noise, those also need to be adequately controlled for. So in the last 10, 15 minutes, I'd like to open this up for questions. If anybody has used layer fMRI or the LayNII package, what's your perspective? What would you recommend? What do you see as the potential for this going forward?
So Andy, you've played around with LayNII a little bit, haven't you?
A little bit, just with their examples, yeah.
How do you feel about the interface and ease of use?
It takes some time to get used to. They do have a very detailed website. Again, they have their own specific brand of humor. So that the analogies they use may click or not. But again, they do provide, like with the figures I was showing before, with kind of an idealized gray matter, white matter boundary. Obviously it works pretty well. You can get these equi-volume, equi-distant maps. But I haven't applied it to an actual data set to try to see how the fMRI activation maps onto these different layers. The interface itself, again, I mean, a lot of it's driven from the command line, which, if you're comfortable with that, tends to work pretty well. They have a large library, a lot of parameters, a lot of things you can choose from. It's kind of mix and match how exactly you want to analyze a specific data set. And they don't give a specific pipeline that's going to work for everybody, which is either advantage or disadvantage, depending how you look at it.
Is it set up to do the segmentation and the layer specification from the fMRI data itself, or is it like from structural images?
Yeah, in the paper they recommend using other segmentation tools. So I think one of them was one of Suma's, I believe, segmentation tools. I don't know about FreeSurfer, for example, but maybe ants possibly. But yeah,
But were they
talk about that in the discussion. Yeah.
But were they using like additional images beyond the activation time series, or were they just taking the VASO data itself and doing the segmentation on that?
I think they were, yeah, taking the VASO data, then doing the segmentation on that, but I'm actually not sure what they used within the paper. I can't remember.
Right.
Yeah.
Thanks. Yeah.
Yeah. Has anybody else used LayNII? Just curious.
I think Max has also played with it.
That's right. Yeah. There are some papers coming out that have used LayNII, and especially visual cortex, for example. That's one of the most obvious applications. How do you see the different layers of visual cortex activating possibly at different times in response to a visual stimulus? You need really high temporal and spatial resolution for that, but that's one of the applications. Beyond that, like we saw, if we talk about the so-called homunculus and the motor and somatosensory areas, and this could also give greater insight into exactly which layers are responsive to what part of the body is being moved or stimulated, for example. So many interesting possibilities opened up with LayNII. I should also mention I'm attending their workshop in 2 or 3 weeks, so have a better idea from the experts exactly some of the answers to these questions. I'm assuming the segmentation is going to be pretty much done for us because it's like a 3 or 4 hour workshop, and I don't think we're going to be segmenting the whole time. Yeah.
Do you guys know if people here at U of M have any, I don't know, scientific questions that could benefit from looking at layers? Asking for a friend. Does anyone in this call have any interest in looking at layer-specific activity? Or
I can start. I don't have a specific question or data set, but I am interested in it. Just as a matter of principle. I mean,
yeah.
I've been moving more towards service-based analysis. That seems like the next logical step. And I think especially with something like functional connectivity, it could be really interesting. Because now you're looking at layers connected to other layers. How does that happen? Like, what does it mean to have one time series in one layer correlate with a time series in another layer? I don't know.
Yeah. I've read some stuff from John Polimeni's group where they were looking at connectivity across layers, and it's like pretty interesting stuff.
Yeah.
But I don't think anyone here is doing that, right? Or interested in.
Not that I know of. This is really okay. This paper was published 5 years ago, but it's a pretty new technique. The principles have been known for a while. People have done some layer fMRI before this toolbox, I believe. But it is very novel technique in a lot of ways. Not many people I know use. I don't really know anybody who uses it. Not personally. So we could be at the forefront,
Well, yeah. I mean, typically the thing that they say is that you need a 7T to do this.
Yeah.
Um, so we have a 7T animal scanner. Um, but I mean, I don't know. I'm currently working on trying to see if we can do have some workarounds so that we can do this at 3 Tesla. And I don't know, I'm getting some hints of success, but I kind of want to know if there's potential collaborations here at Michigan for people who would actually want to do this if I get it to work right. Um,
Any interest from the gallery?
I think the crickets are interested.
Yeah. They're like, yeah. As soon as you said it's going to take a long time to
Yeah.
segment these things, count me out.
Nick, we'll have this working next week.
Yeah, right.
Yeah, I remember seeing a lot of it at this would be a couple years ago now at VSS, the Vision Sciences Society meeting.
Mhm.
So I've seen it used basically just in like V1 and that's it. But
Mhm.
at least in my experience, I haven't seen it used that much in other things, at least as widely as I was seeing at VSS, where there was like
Hmm.
a whole like, cluster of posters.
What sort of tasks were they doing that would differentiate between layers?
Um,
Do you remember?
I think it was like retinotopic mapping,
Hmm.
I think. But this was a few years ago now and in a slightly or pretty adjacent area. So I wouldn't take that the most seriously, but I'm pretty sure it was retinotopic mapping. And I think that there's some of it might have used the what does NSD stand for? The Naturalistic Scenes database. I know that that has a 7T retinotopic mapping
Hmm.
task as part of it.
Okay.
Yeah.
Thanks.
Do you
Do you work with the visual system yourself, or?
I did in grad school, but it was more high level vision. But my question of interest was less about the visual system itself and more about the link between resting state connectivity and task activation within an individual.
Mm.
So the visual system and like the like ventral visual stream is just a good uh I guess domain for looking into that just because the responses are pretty strong within an individual on the task side of things.
Gotcha. Thanks.
If there are no more questions, I think we can wrap it up for today. Just be on the lookout for this. It's a new technique. It's exciting. It's very advanced, obviously. We'll see how practical it is, especially for larger data sets. But keep an eye out for it. And I hope this helped you just understand some of the basic theory and principles behind it. So thank you very much. And I'll see you all next month. Thank you.